Okay, for today, we're going to start doing some molecular phylogenetic analysis for real. Not just conceptual, not with looking at individual cultivated organisms, but we're going to start using it for real. The reality is, and I know you guys have heard this before, and I've told you this before, those lights are not good. Here we go. That most organisms in the wild don't want to grow on plates. How many of you have experienced this in lab? Right? When you think about it, those organisms have been living and their ancestors for 3.6 billion years in the wild, and you're expecting them now to grow on a little agar plate. Not anything at all like the natural habitat of the organism. And so the fact that this ever works is amazing. But the reality is it doesn't work more often than not. The often quoted number is less than 1%. People would say that less than 1% of microbes have been cultivated. I think if you look at the statistics of cultivated organisms versus sequences that were fished out in the way I'll describe here, you can make a pretty good argument that 1% is orders of magnitude too high. That is probably closer to somewhere around 1 in 10 to the 6 or 1 in 10 to the 8 cells that are kinds of cells that are in the environment can be cultivated in the lab. You'll do this experiment in the next 2 weeks in lab, and it's uncommon for any one student in class to get 1% of what they can see in their Winograsky column actually growing up on a plate. The average number is somewhere around 1 in a million. So, how do we get a handle on these organisms, the ones we can't grow? And that is the question. What's the handle? How can we get something to grab onto so that we can investigate them? Otherwise, they're just specks that you see under a microscope. Well, molecular phylogenetics is one way to do this. And so how does this work? If you look at this graph, this should be a little familiar to you, because part of it is what you've done in lab. You start with a microbial sample. In most cases, this is a pure culture. You do some kind of extraction to give you DNA. We kind of circumvent this by putting cells directly in the PCR reaction. You run an amplification to amplify up the ribosomal RNA or any other sequence that you might want to use as a handle. You do some, or you send it off these days to get a sequence, and you get some data back, a string of letters. And from that, you can do a phylogenetic analysis and find out what it is. But you don't have to start with a pure culture. We've already talked several times about cases where you had an organism where you couldn't grow it, but you could still do this because you could isolate the organism physically, even though you couldn't grow it. And so one case of this was Coraceum, where they could isolate the organism in an enrichment culture and physically get rid of everything else using treatment with detergent. We also talk about this in terms of the bacterium symbiont in aphids, where you have this vitamin amino acid building bacteria in the aphid, which of course drinks sap. And you can physically dissect the bacterium, isolate bacteria from that, and amplify ribosomal RNA and find out that that organism is a particular kind of creature, in this case, Buchnera feticola. And people have done a lot of this kind of stuff. We're going to talk an example next time, Monday, where people have done this on a deep-sea hydrothermal vent animal that uses bacterial endosymbionts for energy. But you can actually do better than this. Because maybe you're not interested in particular organisms, but maybe you're interested in the community. What are the members of a community? So in this case, you might start with a microbial sample that's a little more complicated. 2 scrapings, an infection site, filtrate from open ocean water, bits of a microbial mat, soil. Isolate DNA. Now in this case, you're not going to get a single sequence amplified, right? you're going to get your DNA may look like a band because all the DNA fragments are about the same size, but that population of molecules is heterogeneous. It's going to be a mixture of different sequences. And if you've done the process more or less correctly, the relative ratios of those sequences ought in some way to reflect the abundance of the organisms in the original sample. Now, there's a variety of reasons why that's never actually true, but the hope is that it's at least qualitatively a reflection. At this point, then, you've got to separate those out, because if you try to sequence them directly, you'll get a commission sequence that you can't interpret. So you've got to separate them out, and depending on your sequencing technology, this either means doing reactions that work on a single molecule, or just cloning them into a plasmid vector of some kind and separating them out to different colonies. Then you sequence a bunch of them. And if you sequence enough of them, hopefully that your population of sequence data, again, represents the microbial sample that you start with. Now, the problem is very often is that once you get sequence data, how do you prove that that has anything to do with your original sample? And the common way to do this is to take your sequences or collections of those sequences and design oligonucleotide probes that are complementary to a variable part of the ribosomal RNA. To that oligonucleotide, you attach covalently a fluorescent marker, fluorescein, or acridine orange, or Texas red, something like that. And then you use that as a probe to go back to a copy of your original sample and probe it using what's called fluorescent in situ hybridization. So you take the sample, you fix it in glutaraldehyde, it kills all the cells, locks them into place. You permeabilize the cells with xylene, and then you mix some of this probe in, and it soaks into the cell. The oligo will anneal to the ribosomal RNA if it's a match, and then you wash the stuff away, and if the cells glow, then it's a match to your sequence. You use the probe against the ribosomal RNA because if you probe something in the DNA, there's only one or a few copies in the DNA, and there are thousands of ribosomes. So we're going to do a bunch of this. You can use this approach to identify microbial groups in a population. You can use it to count them. So if you've got a probe against a particular kind of organism, you can use fluorescent in situ hybridization to light them up in a sample, and you can go through and count them. You can look at their morphology. You can watch an enrichment culture and see if the organism you're interested in is getting more abundant over time. We'll talk about an example of that today. If you design your probes intelligently, you can make them specific for different phylogenetic groups, and you can probe a bunch of samples looking at whole groups of organisms. For example, you might imagine a bacterial-specific probe and an archaeal-specific probe, and you could probe an environment and look at the ratio of these two creatures. And for identification, that's kind of the easy one.